Hello again, and in this video, I'm going to be talking about color vision. Now, before I get into color vision, I just briefly want to go over, photoreceptor cells and the two main types we have. So firstly, we have rods. So what rods are, they are, the monochromatic, which means one color type of cells where, they're mainly the night, they're a lot more abundant. And then we have, the cones, and cones are actually what we use to see color. Cones are they're not monochromatic as that's what is we use, like I said, to see color, and they're in less abundance, but they're a lot more sensitive. So now looking at specifically, different types of cones. So cones, there's three different types of cones. There's short wavelength cones, S cones, medium wavelength cones, M cones, and long wavelength or L cones. So what happens with these cones is that they're named really after their absorption peaks. So short, obviously, are close to like 420 nanometers wavelength, whereas medium are 531 around, and long are about 558. So with these short cones, we kind of see that bluish color. That's where they peak around. With these medium cones, they're kind of the closer to the yellow scale. And then with these larger cones, you see more of like the red portion of it. And the rods kind of fall in between, the S cones and the M cones. So, with many individuals, there's variability. The fact of the matter is that the variability in the ratio of the L and the M cones is, an indication that people kind of perceive different colors in different ways, which is actually, very interesting. But, how is it that these different cones leads to different cognitive processes of understanding, or differentiating one wavelength of light from another? So what we see is with the S cones, the blue cones, the M cones with the green cones, and the large cones with the red cones, they have different opsins and they interact with different wavelengths of light. So the lower wavelengths of light let's say the S cones, they will interact with blue light, and then this will cause a transformational change in their, as I said previously in previous videos, with their cis retinal to form to an all trans retinal. So depending on the wavelength of light, this will actually interact with different opsins depend on which wavelength of light activates which opsin. And what's actually very interesting is there's different levels of activation of these different opsins. So if you have total activation of all the opsins, this will lead to white light, or just white being perceived. Whereas if it's no observation, no opsins, this will lead to black being perceived. However, if there's different combinations of them, then this could lead to different colors being perceived. So if we have, let's say, the 100% of the S opsin being activated and 100% of the M opsin being activated, but let's say there's 0% of the L opsin being activated, this could lead to a light bluish tint being seen. So it's just interesting to see how depending on which opsins are activated, this will lead to different color perceptions. So this, again, was just an overview on color vision and how our brain registers different colors based on the activation of different opsins. Thank you.